Genetically modified foods are foods that have been genetically engineered to display beneficial traits, including resistance to pests and chemical treatments. With the technology that we possess, we can make certain traits on the planet or animals more pronounced. They fall into the larger category of genetically modified organisms, or GMOs, which are also used in many other fields of studies like medical research. Since the early 1990s, GMOs have been put into many of our food sources coming from plants, and the possibility of genetically modified animals are also being explored. Although farmers have been using selective breeding to achieve desirable traits in plants and animals for hundreds of years, this process is nowhere near as efficient and precise as genetic engineering. Essentially, this process involves taking a desired trait from one organism and then transferring it directly into another. To make this all possible, these plants are modified using recombinant DNA technology that William will discuss.
Several advanced genetic engineering techniques and technologies are required for the production of genetically modified foods. Evidently, the first step in this process is finding a desirable trait in an organism that will be transferred to the target organism. The nucleic acid sequence that codes for this trait is isolated using comparative methods. Subsequently, this gene is incorporated into the genome of the soon-to-be genetically modified plant. One common method of accomplishing this involves the use of a device called a gene gun, which shoots metal particles coated with DNA directly onto the plant's cells. Alternatively, the gene can be introduced via a bacterium, which naturally invades the seeds of a plant and alters SLE. The newly developed plants must be closely monitored to ensure that the desired phenotype is being produced. Sometimes, automated machines are used to track the growth of plants and determine optimal growth conditions.